guys, our lesson today will be about photosynthesis, IB Diploma Biology 2.9. The essential idea is the main aim of photosynthesis is to transform light energy into chemical potential energy that can be used by the organism. Photosynthesis is the production of carbon compounds using the light energy. Carbon compound includes all forms of high energy foodstuff, including carbohydrates, proteins, fats, and um, amino acids, DNA, and so on. Here, the green plant is going to absorb light energy and transform it into high energy foodstuff that we call them potential energy, chemical potential energy. The simplest molecule produced in photosynthesis is glucose molecule, but from glucose the green plant can synthesize more complex foodstuff. Oxygen is produced by photosynthesis from photolysis of water. Photosynthesis occurs in two main stages inside the chloroplast of the plant cell. We have studied before the structure of the chloroplast. You know, it has stocked of membranes, we call them grana, and between the grana there are spaces, these spaces where the chlorophyll molecules stay. And we call the group of grana thylakoid. The first stage of photosynthesis, it depends on light. We call it light-dependent reaction, and this occurs inside the thylakoid membranes or in the grana. It starts with photolysis of water. Water molecules, when they are exposed to sunlight, are going to break down into oxygen and hydrogen. Hydrogen will be taken by hydrogen-carrying compounds like NADP, nicotinamide adenine dinucleotide phosphate, while oxygen will be released into the atmosphere as a byproduct. Also, this energy will be consumed by molecules of ADP to produce ATP. ATP is used to provide energy for the second stage of reaction that doesn't depend on light, and we call it light-independent reactions. Energy is needed to produce carbohydrate and other carbon compounds from carbon dioxide. So the second stage in photosynthesis, we use energy from ATP. We use hydrogen carried on hydrogen carrier like NADP to transform carbon dioxide into carbohydrate, and we call this process carbon fixation. This process is done by a scientist named Kelvin, so sometimes we call the process of carbon fixation Kelvin cycle. The enzymes which catalyze the process of carbon fixation, we call it ribosco, ribulose, biphosphate carboxylase. This is very important enzyme because it catalyzes the first stage in the light-independent reaction where carbon dioxide will be fixed into high-energy food molecules and like carbohydrates. Energy is needed to produce carbohydrate and other carbon compounds from carbon dioxide. You can see here, this is a summary for the whole process. Light falls on the thylakoid membranes of the chloroplast. Water undergo photolysis into oxygen and hydrogen. Hydrogen is used to transform NADP into NADPH, while some of the energy is used to create ATP from ADP. All of those are considered as raw material that will be used in the light-independent reactions. Light-independent reactions happen in the stroma of the chloroplast where ribosco and the other important enzymes exist. The first stage in the Kelvin cycle is fixation of carbon dioxide using hydrogen carried on NADPH and using energy that was stored in ATP molecule. As a result, sugar molecule is produced. So we can say that photosynthesis generates chemical store of energy in the form of carbohydrates. Light-dependent reaction use light to produce ATP and to split water that we call photolysis, making hydrogen ions. While the light-independent reactions, it uses ATP and hydrogen ions to fix carbon dioxide making glucose. Starch and cellulose are polysaccharide molecules found in plants. Starch is the chemical store of energy, and cellulose built up the plant cell wall. So once the plant produces monosaccharide like glucose, further processes should take place in order to convert glucose that we call them monomers into more complex sugar polysaccharides like glucose and like starch and cellulose. Here is a summary for the process of polymerization where the monosaccharides convert into polysaccharides. Here is the summary for the condensation reaction. This reaction, we call it condensation. Water molecule will be removed from two sugar molecule in order to join them together by a bond. We call this bond glycosidic bond, and the water molecule will be given out. Because green plants chlorophyll molecule absorb light energy, so we need to have a closer look for the visible light. Visible light has a range of wavelengths with the violet as the shortest and the red is the longest. Normally, the visible light has range from 400 to 700 nanometers. 400 is for the violet region and 700 for the red region in the spectrum. The spectrum is considered as electromagnetic radiation that reaches the Earth's surface, and here is the frequency of the radiation. Chlorophyll absorbs red and blue light most effectively, so it's going to reflect the green light. So when the sunlight, the visible light, fall on the photosynthetic pigments inside the leaf, photosynthetic pigment is not only chlorophyll, but chlorophyll is the main photosynthetic pigment. Even we do have two forms of chlorophyll, chlorophyll A and B. In addition to that, there is xanthophyll and other photosynthetic pigments. Those pigments, they are going to absorb the visible light in the blue and red regions. These are absorbed, while the rest will be reflected. The reflected color appears green. This is why you can see the leaves green. This spectrum, we call it action spectrum. Action spectrum here, you can see the maximum absorbance of light. Green plants or chlorophyll, the maximum absorbance in the violet region, violet blue region in a wavelength 400, and another peak at range about 750. Okay, the green yellow region is not absorbed, and this is the part which is reflected. So action spectrum shows you the range of wavelight which can be used in the light dependent reactions. Whereas the absorption spectrum for the chlorophyll, it does show the level of absorption of light by the chlorophyll molecule or by the photosynthetic pigment. The rate of the wavelength is here, and you can see also that there are two graphs here. The first one is for the rate of absorbance, which is maximum in the violet and red, and the second one is for the rate of photosynthesis, which is also very high in the 400 region and 700 region of the spectrum. Here, when the two graphs overlap, both of action spectrum and the absorption spectrum, it just to give you an idea that the chlorophyll is the most important pigment or most important part in the photosynthetic process. This will be more clear if we use artificial light source to prove which light sources are better absorbed by the chlorophyll molecule. You can see here the absorption in the blue region, and here the absorption in the red region that happen very effectively. You might be asked this question: why do leaves turn brown in the autumn? This because in the autumn, when the plant is not photosynthesizing, because the light energy is not so high, the chlorophyll will be removed from the leaves. As a result, other pigments became dominant: red pigments, orange pigment, brown pigments, and so on. This is why the plant looks different colors in autumn when it's not photosynthesizing. And it's because, of course, if there are other pigments, they will be having other peaks of absorbance other than the red and blue regions that the chlorophyll can do. Chromatography is a very important process that can be used to separate the pigments of inside the photosynthetic pigments. You know how the chromatography process can be run. Chromatography is a method of separation different pigments molecule based on their solubility and molecular mass. Here, and also, of course, it depends on the type of the solvent used. In this experiment, we are going to crush the green leaves and mix them in acetone or ethanol, and then you are going to carry out filtration process, and you will get the filtrate. Uh, Heat the filtrate on the basin, on the hot basin, until it evaporates and becomes dry. And then just put 1 drop of ethanol in a petri dish or watch glass and put it here on the basement line on the chromatography paper. Put the solvent, make sure that the level of the solvent does not exceed the baseline. As the solvent takes the spot with it, the pigments in the leaf are going to separate into their components. So we can know how many pigments and what are the colors and types of pigments exist in the green leaf. Now we are going to discuss limiting factors that affect the rate of photosynthesis. Photosynthesis is controlled by many factors. The main factors are temperature, light intensity, and carbon dioxide concentration. They are all limiting factors. If we are asked to uh, explain or investigate the effect of any one of those factors, you need to keep all other factors constant, and they should be in very high concentration. Suppose you are investigating the effect of carbon dioxide concentration on the rate of photosynthesis. This means that you are going to vary only the carbon dioxide concentration. And you need to keep everything else at maximum. So keep the temperature at optimum for the green plants to photosynthesize. Light intensity, put plenty of light intensity. And also you can put the required minerals for plants and stabilize all the conditions required, and then vary only the percentage of carbon dioxide. So now you can monitor the effect of carbon dioxide on the rate of photosynthesis. It's clear from this experiment that as the carbon dioxide concentration increases, the rate of photosynthesis increases up to a point we call it plateau or level off. At this point, the carbon dioxide is no longer limiting for the rate of photosynthesis, as the rate of photosynthesis now have reached its maximum efficiency, and it will not be affected anymore by the carbon dioxide. So if you need at this point when the rate of photosynthesis is stabilized, if you need to increase the rate of photosynthesis even more, you need to look for any other factor to change it, but not for carbon dioxide. This graph shows you the effect of light intensity on the rate of photosynthesis. Okay, so you can put the plant at different distances from the light source and look for the rate of photosynthesis and plot your graph. But how we can measure the rate of photosynthesis? The rate of photosynthesis can be measured by either the appearance of starch in the plant, but more easily is to find the volume of oxygen produced per unit time. So at every point in the light intensity measure the rate of photosynthesis by measuring the volume of oxygen produced. In this way, you can monitor and plot your graph. Here, the effect of temperature, you can see that the curve is very similar to the effect of temperature on the enzyme activity, because temperature controls the light independent reaction that mainly depends on enzymes. So as you know, as the temperature increases, the enzyme activity increases, so the rate of photosynthesis increases. But up to an optimum temperature, any increase in the temperature above the optimum will not affect the rate, but the rate is going to decline. Why the rate decreases? We can say that because at very high temperature, the enzymes denature, and then the light independent reactions will not take place as effective. Think here, how would the pH affect the rate of the photosynthesis? pH is one of the factors that affect enzyme activity, right? Because photosynthesis is an enzyme-controlled metabolic pathway, so all the conditions that apply to enzymes will apply for the rate of photosynthesis. The rate of photosynthesis is at the peak when the pH is at its optimum for the plant. Moving away from the optimal pH, up or down, before or after, will lead to denaturation of the enzyme because the hydrogen bonding holding the secondary protein structures will be broken, and then a change happen in the active site, so that no longer fits the substrate and the reaction cannot occur. To measure the rate of the photosynthesis or to design an experiment to investigate the limiting factors, first you need to show how you are going to measure the rate of photosynthesis. Here we are measuring the rate by the change happen in the oxygen volume over time being observed. Oxygen production, how to find the rate of oxygen production. You need to, you may count the bubbles of oxygen produced per unit time, or you can collect the gas in a gas syringe and you get the volume. You can get a more quantitative data by measuring the volume using a gas syringe. Another indirect way that can be used to measure the rate of photosynthesis rather than using the carbon dioxide, that is the change in the biomass of the plant, because as the plant photosynthesize is going to make more sugar, more carbohydrates, more plant cells, so the biomass of the plant is going to increase. But remember that every time you are measuring the biomass, there are so many errors in this experiment because it's a wet mass, not a dry mass. It will be more accurate if you measure the dry mass of the plant rather than the total biomass. This experiment is investigated to find the limiting factors that affect the rate of the photosynthesis. We got an Elodia plant or a water plant. A water plant is here, and you put a source of carbon dioxide like sodium hydrogen carbonate, expose the plant to the light source. Let's answer this question. Why are the following procedures necessary? Boiling and then cooling the water before the experiment. We boil the water to make sure it's free from pathogens, free from any factors that might alter the rate of photosynthesis. But why we cool it down again? We cool it down to keep the temperature optimum for the enzyme activity that are very important in the light independent reaction. Keeping the water at 25 degrees. So this is an optimum temperature for enzymes in plants, and highly illuminating it. Light is very important because light is a very important limiting factor for the rate of photosynthesis. Counting the bubble. So you need to repeat counting the bubbles until several consistent counts have been observed. This is to make your experiment more reliable. What other factor could be investigated using bubble counts? How would you design the experiment? We can use a bubble count in any reaction which involve aquatic plants. We can use it to investigate the effect of carbon dioxide concentration or the effect of light intensity on the rate of photosynthesis. Also, you can get some different plants that have different surface areas of leaves and compare the rate of photosynthesis. How can you make your experiment or your measurement more accurate? To make your experiment more accurate, you need to find a more quantitative way to measure the light intensity, to measure the oxygen produced. Changes to Earth's atmosphere, oceans, rock deposition due to photosynthesis. Early Earth's reducing atmosphere contained very small amount of oxygen. The first prokaryotes emerged around 3.5 billion years ago, but it wasn't until about 2.2 billion years ago that oxygen level rose to 2%. Because people suggest that when Earth started, when life started on Earth, the atmosphere was mainly made of carbon dioxide and ammonia or nitrogen compound. But prokaryotes very small green bacteria have been arisen, and as a result, they start to make photosynthesis. It took like more than 1 billion years until the level of oxygen became 2%. We call this process or this event great oxidation event. The changes to Earth's atmosphere, ocean, rock deposition due to photosynthesis. As a result of the photosynthesis, we would like to thank the photosynthesis because it has been observed that the presence of photosynthesis and the increase in the rate of photosynthesis lead to very high rate of increase in the oxygen in the atmosphere, and also dropping in the level of both methane and carbon dioxide. You know, methane is a very toxic gas, and also carbon dioxide has very harmful effect on the atmosphere and on living organisms. And this was corresponded to what we call Earth's first glaciation. Also, scientists thought that the changes to Earth's atmosphere has resulted in corresponding oxygen of dissolved iron in the ocean, allowed iron ore to precipitate, forming bands across the seafloor and providing source of iron and steel to the day. So also we would thank the photosynthesis for having iron, and you know steel manufacture and iron is very important metal in the recent industry. These graphs show you the changes to Earth's atmosphere, oceans, rock deposition due to photosynthesis. You can see that here the changes that happen in the rate of photosynthesis over time. Oxygen rise to 20% now of atmospheric concentration, and this happened around 600 to 700 million years ago. And this was accompanied by multicellular speciation or arising of millions of species of living organisms. Thank you guys, we are done with our lesson.